Welcome back to the course Fundamentals of General Immunology. We are in Module 2, Systems Level Organization of Immunity and Immune Recognition and Threat Detection. So this is our lecture number 3, Coordination between Cellular, Molecular and Tissue Level Immunity. So let's understand a very basic of molecular biology, which we generally call central dogma. What central dogma says is that we have these molecules which are called DNA. These are genetic information. This can convert into second messenger called messenger RNA, and then this messenger RNA can in turn convert into protein, which can be actually the functional unit of a cellular system. So basically the DNA to RNA conversion is what we call transcription, and RNA to protein conversion is what we call translation. And post discovery of reverse transcriptase enzyme from retroviruses, we also understood that there is a possibility now that we can convert RNA back into cDNA by an enzyme called reverse transcriptase. So this is what we call reverse of central dogma or Teminism, because of the name of the scientist Temin in Baltimore who discovered this phenomena. Also, the DNA can replicate themselves back to the same molecular DNA by the process of replication. So if you grossly look, look into this phenomena, this is very fundamental aspects of molecular system. So suppose if we draw a cell, and if we know that these are the nucleus, and so let's see, this is cytoplasm, this is nucleus, and we have a DNA here. So the DNA dependent RNA polymerase can bind to the DNA and can give rise to an RNA, and these RNA can transport into the cytoplasm and can stick to something called ribosome, which is a translational machinery to produce a protein. And this protein is actually doing all the function, including some of the cellular process. So these proteins are actually doing some function, including the cellular processes, and also these protein can be secreted out and do certain other functions as well into the nearby tissue. So what is basically understood in the molecular sense that these proteins are actually the functional unit, and post production into the cytoplasm they can start doing their functions. Now in immune system or immune cells, when we are trying to understand the immune cells, what we could see that these immune cells are actually producing lot of different chemicals which are collectively called as cytokines. So cytokines actually are specific molecules which are produced by an immune cell in order to do functionality of a cell. Now this cytokine could function in three different aspects. Either they can go to the distance place through blood circulation and do a function what we call endocrine function. So these are the protein which can go to the further in distant organ to do some function which is endocrine in nature. This cytokine can modulate its own producer cell by something called autocrine function. So the cytokine may do an autocrine function which is largely understood for the immune cell, or cytokine can also do modulation in the neighboring cell population, what we call as a paracrine function. So in general, these proteins which are produced by a very orchestrated molecular phenomena of this central dogma could lead to activation, downregulation or modulation of the immune system or immune cells. Now these chemicals, which are just one example we are considering here, is these cytokines which are produced by the cells. Now if you grossly look into the term, cyto stands for cell and kine is the kinetics. So basically these are the chemical which can give kinetics to the cell, and therefore this can function either to modulate themselves. So these are called autocrine function. They can go to the neighboring cell population and do an activity which is called paracrine function, or they could migrate to the different cell population or the distant area to do an endocrine function. So these are the three functions of the cytokine which we need to look into in a different way. So let's move on and try to see that what are the other things which a cytokine do. So cytokine could do a signaling. So these are the general schemes which we are talking about. So suppose these are the cytokines, which are actually what we are calling right now here is the signaling molecules. So they can bind to something on the cell surface. So if you see here, so this is the cell, and this is the cytoplasm of a cell, and these are extracellular space. So we have some proteins which are expressed on the cell membrane. So these are cell membrane, and the cytokines which are produced from a immune cell or any other cellular type in the human body can bind to this protein, and binding of this cytokine could activate this inactive protein into an activated protein. And this activated protein can further do a lot of biology which are very important for the immune regulation. So what we are trying to understand, that once the protein is produced following the central dogma, the protein can function a lot of other aspects using the immune cascade. One such phenomena here, we are talking about the enzyme activation on the surface. So the cytokine can bind to the receptor which are present on the cell membrane. Remember, these are glycosylated protein. So these proteins are heavily glycosylated. That means there is a post-translational phenomena that can add the sugar molecules into the protein to make it functional. And because of that sugar molecules, these proteins are dragged into the cell surface. So once the protein dragged onto the cell surface of a cell can bind to the cytokine, they can activate themselves, and these activation can lead to further downstream cascade that could lead to the immune regulation or immune functionality of a cell. So this is a general scheme. The second is, say we'll just take an example of a pathogen, for example. So suppose this pathogen is entering inside the host body. There are sensors which we'll discuss a little later. So these are, suppose, the antigen presenting cell or the cells which can encounter the pathogen. So they have certain kind of molecular structure on their cell surface which we call sensors, and this sensor can sense these pathogens. And post activation, since we understood that this binding can lead to production of some chemicals, and these are called cytokines, and these cytokines will be produced into the system. They can activate a separate cell part type, and we are trying to understand here some of the immune cells such as lymphocyte. And remember, this we are talking as a general scheme. So this could change, this could modulate. There are a lot of other phenomena that could happen in the immune system, but this is just a general scheme. Further, this lymphocyte can do a second round of cytokine replication or cytokine production, because earlier it was produced through this antigen presenting cell which can bind to the pathogen through these sensors. Second order is through this activated cytokine produced from this lymphocyte, activated lymphocyte, and these cytokines post production can do an effector function, and this effector function could basically neutralize the pathogen. So if you see grossly, this is the cascade actually. So the pathogen to sensor to cytokine to lymphocyte to second order cytokine, effector, and finally response towards the pathogen. So ideally, these are the general scheme which we need to understand when we have any molecules which are secreted through the immune cells. All right. Then we also try to understand a very unique phenomena that is happening in the human body, and that's called diapedesis. So diapedesis is nothing but oozing out or movement of a T cell population basically, or B cell population, or a lymphocyte, or a blood cell from the blood vessel towards the site of lymph node or an extracellular space. So what's happening, that this is a very, very orchestrated phenomena once again, because all the immune regulations are very orchestrated. So if we look into that phenomena in detail, the first event is called rolling, and what rolling stands for, that this lymphocyte or the immune cells are rolled over this high endothelial vanules, which are the lining of the vessels. So this lining will have a lot of selectin molecules. So there are a lot of proteins which are present in this endothelial surface, which are called selectins, and these surface receptor which are present on this cell population can bind to the selectin and just do a rolling phenomena. So this rolling phenomena is again a very important immune regulator response in the human body. There are other antigens which are other surface receptors which are present on the surface of a lymphocyte or immune cell, such as the LFA. If you look into the full form here, the leucose lymphocyte function associated antigen 1. Actually, it's an heterodimeric integrin protein that's expressed on the surface of immune cells, some of the immune cells. And then the endothelial lining, apart from other selectin, they can also express the chemokine ligands, such as chemokine ligand 21 and also chemokine ligand 19. So there are other chemokines as well. We are just looking into some examples here. So these chemokine ligand 21 or some other ligand can bind to that chemokine receptor. For example, here we are trying to understand chemokine receptor number 7 that can bind to chemokine ligand 21, and this rolling phenomena is getting a second shift towards coming out, giving an access of these cells to the endothelial vessel towards outside or the extracellular space. So this binding of CCR7 to CCL21 can start a separate cascade which is called adhesion. So once they activates, then we start an adhesion. Adhesion stands for sticking of the cellular system towards something called ICAM1, which is an intercellular adhesion molecule, which are also expressed on the surface or lining of the endothelial vessels. So this ICAM1 can bind to LFA1, and because of this binding or adhesion, essentially these cells are coming into this outside space, which is here exampled as lymph node, and this cell can further do an activity. So basically the cells of the immune system are generally in the blood vessels, and if there is an infection here somewhere in the extracellular space, these cells has to come out in order to do an effect. Therefore, this diapedesis phenomena is again fundamental phenomena of the immune system, where the rolling activation, adhesion, and then the release of the cells into the extracellular space is what collectively called as diapedesis. So again, since we are trying to see how these molecular details and the cellular system are integrally enabling the system, so we could look into that and we can understand that we have cell, for example the T and B lymphocyte or the neutrophils or other basophil, where we discussed in the last class that these are the cells of the innate immune system or adaptive immune system, in addition to some of the other cells such as the plasma cell or the mast cell. Now these cell population are having lot of integral gene expression which could lead to production of different kinds of protein, and these different proteins can modulate many function of the immune cell, which we'll slowly learn into when we go deeper into this course. But ideally we should understand that this molecular orchestration of central dogma is highly regulated. Gene expression of an immune cell is highly regulated phenomena. All right, so when we talk about the cellular system, every cell in a human body is having a finite life, and also the cell population can exert or exhibit certain response towards the external stimuli. For example, we are listing here two phenomena. One is called apoptosis and the other is called necrosis. Now these two are actually the phenomena which is called cellular death. Now this cellular death, since although it is a cellular death, but there is a difference between these two phenomena. Suppose if the cells are stressed, and this stress could be because of multiple reasons. For example, if there is a toxin secreted into the body system, or if there is any kind of an enzyme which are produced into the cellular system, that could lead to the stress condition in a cell. Now there are two possibilities, that there could be a cell death where the cell could recycle the stuff for the betterment of the host itself, and that's called apoptosis. In the broader sense, it is called programmed cell death, which is programmed. There is no much harmful effect associated with the apoptosis. But if there is a harmful toxin or any other moiety which are harmful for the body, then this phenomena is actually called necrosis. We'll see the difference between apoptosis and necrosis in the next slide, but at least for time being, one has to understand that apoptosis is a phenomena of programmed cell death. Cell is doing this phenomena in order to recover the molecules for the betterment of the health and all. Necrosis is also a cell death, but this is actually a harmful effect because these are producing lot of chemicals which are collectively called as damage associated molecular patterns, DAMPs, DAMPs. So ideally this necrosis is actually a harmful cell death where the cell is dying because of harmful or toxic environment which is present in outside system. So let's try to understand what are the differences between apoptosis and necrosis. So as I told earlier, that apoptosis is called programmed cell death. So for example, if there is a lifespan of a cell around 20 days, after 20 days this cell has to undergo recycling, and for that reason this cell which produced the whole bunch of phenomena, after 20 days has to die, and this phenomena is coming under programmed cell death. While in necrosis it is kind of a cell death which is accidental, and that could be because of entering of any foreign particle or molecules. In terms of cell size, there is a shrinkage. That means the cell is reduced to its size so that to do a phenomena of apoptosis, while in necrosis we have swelling. So it's just opposite to that. We have a shrinkage, here we have a swelling. That means cell size become increased. The nucleus is again a very fundamental organelle of a cell type. So there could be a fragmentation of the nucleus which we call karyorrhexis, and then here we have a pycnosis which is actually leading to karyolysis. That means here the nucleus becomes fragmented into smaller fraction, while in case of necrosis the nucleus is lysed into deeper fragments. That means there is complete destruction of nucleus here, while in this case the nucleus is getting fragmented into smaller pieces. The membrane is intact. That's very important here. Since the phenomena is useful for the body, so the membrane is intact here, while here these are all ruptured. That means the content of the cellular system is just out. What we have understood that this could lead to the damage of the neighboring cell population as well. But here the things are intact because membrane is intact here. There is no inflammation associated with this apoptosis phenomena, while in case of necrosis we do have inflammation associated with it. The cause is because of the development, injury or signals that could lead to apoptosis, while here any sort of trauma, toxin or hypoxia. Hypoxia stands for the lower oxygen concentration in the cellular system. So this could lead to necrosis, while these are kind of an accidental death. So these are always related to any toxicity associated with the cell. Lastly, very important, this is ATP dependent process. That means there is an expenditure of energy. So this is not an sparing process. The cell has to spend a lot of ATP. So this is energy expenditure process, while this is ATP independent. There is no involvement of ATP here. So what we could infer from here that there is an involvement of mitochondria for sure, because the ATP is produced and utilized here, while in this case there is no involvement of ATP. So this is just a passive process, while this is an active process, apoptosis. All right, so we will move on and we will try to understand another important cellular process. So in this whole lecture we are trying to understand what are the cellular events that could modulate the immune system. So first we understood what are the cytokines, how they are modulated to the receptor, how this cytokine can modulate and activate the cascade, and also the cellular process such as necrosis and apoptosis. And there is another phenomena which we will discuss here called autophagy. Autophagy stands for, auto means self, phagy means eating. That means cell itself eating themselves. So what it stands for, when the cell identify, this is a starvation related phenomena. So this is a phenomena which cell does in order to maintain the homeostasis. That means if there is a starvation, if there is a deprivation of chemicals or say metabolites such as glucose, energy driven processes, anything which are having a starvation condition in the cellular system, we do see that there is something called phagophore, which is formed in order to squeeze this these molecules which are there in the cellular system to make an autophagosome. These are produced inside the cytoplasm. Then post production of this, there are a lot of markers which are present in this, so one can easily identify that a cell is undergoing autophagy or not based on the surface marker which are present on the cell surface of autophagosomes. These autophagosomes are then mixed with this lysosome, which is containing lot of hydrolytic enzyme into it. So these lysosomes are getting fused with the autophagosome, and then this autophagosome mixed with lysosome can start degrading, and further it can throw out the content for recycling. So basically what is happening that this autophagy phenomena, which starts because of the starvation, can do lot of recycling phenomena, because essentially the cell identify themselves in the starved condition, and to just maintain the normal homeostasis or physiology of the cellular system, they can do this autophagy phenomena. This is again a very orchestrated phenomena happening in case of human body. All right, so we will try to understand two other terms here. That's called pathogen associated molecular patterns, which is called PAMPs in short, and then we have damage associated molecular pattern, DAMPs, which we just discussed. These are the molecules which are produced post necrosis. So to understand these two important proteins, or these two important entities which are there in the system, we need to understand how they are originated, what are the evolutionary purposes they have, what are the key examples of them, and what are the primary sensors which are there. That means these are some of the things which the body can sense, or the cell can sense actually. And this, for example, these are pathogen associated molecular patterns. So what could be the origin of this? The origin could be any microbes, for example viruses, bacteria, the fungi, parasite, any allergen or anything. So these are exogenous in nature. So once they are entering inside the body, they are actually having certain signature on their surface which suggests that these are pathogen associated molecular pattern. While DAMPs, which are damage associated molecular pattern, is coming from endogenous. The host cell itself is producing post necrosis. So this is our own molecules which are exaggerately produced in response to a necrosis phenomena. That means these are our own thing which are not useful or harmful when it comes to outside system. So these are to detect. So these structures are actually present on the surface of the pathogen, while we have this TAM which could lead to the production of these molecules are because of the cell death or the tissue trauma. The key examples which we need to discuss is some of the things which are present on gram-negative bacteria, and these are called lipopolysaccharide. These are exclusively present in gram-negative bacteria. We could also see that there are viruses which are having a genome either in the form of RNA or in the form of a DNA. We never see that viruses are having both the things together. So either they have RNA or DNA. The RNA and DNA could be of different varieties. For example, the DNA could be single-stranded DNA or double-stranded DNA. RNA could be single-stranded RNA, double-stranded RNA. Or the RNA, single-stranded RNA could be either positive polarity or the negative polarity. That means the positive polarity is ready to produce a protein. These are like a messenger RNA 5' to 3', while negative RNA is 3' to 5', which has to convert back into 5' to 3' in order to do protein synthesis. Then we have CpG DNA motifs. These are also critically pathogen associated molecular pattern. That means these are the molecules which are coming from outside. So these are exogenous in nature. They are not produced by the human system. While there are things which are produced by the endogenously host, and some of them are the high mobility group box protein, HMGB1, ATP, adenosine triphosphate, uric acid, heat shock proteins, HSP in short they are called, and these are also very critical protein useful for the maintenance of homeostasis. But sometimes these are produced in excess, and these excess amount can lead to inflammation or inflammation like conditions in the cellular system. So once we understood that the pathogen associated molecular pattern or the damage associated molecular patterns are produced either by exogenous or endogenous way in the human cell, we could also see that there are receptors which are present in the host cell, and some of them are called toll-like receptor. These are again very important. We will see that in our future classes. These are the sensors of innate immune response, toll-like receptor. These are discovered in Drosophila initially, and there could be multiple TLRs. We are just giving an example of TLR4 and 3 here. So lipopolysaccharide can be detected by TLR4, and also the retinoic acid inducible gene 1, RIG-I like receptor or RIG-I like receptor. So retinoic acid inducible gene is also a primary sensor for pathogen associated molecular pattern, while in case of damage associated molecular pattern, we have something called receptor for advanced glycation end products. This is called RAGE, which is again the sensors of detecting damage associated molecular pattern, and also TLR. Some of the TLRs can also sense this damage associated molecular pattern. And finally, we have this NLRs, which are called nod-like receptor. We will gradually go in detail about all these molecules which are either sensor or also the content of molecular associated pattern, because the binding of molecular associated pathogen associated molecular pattern to the receptor can activate the whole bunch of cascade, which could lead to neutralization of the exogenous antigen. While in case of DAMP also the same thing. Once the DAMPs are produced, the cellular system can find out some of the receptor can bind. So there could be always a binding of a molecule with the cellular partner. Once there is a binding, then there is a whole bunch of cascade which can be activated. Once there is an activation, then we could see that either of the effect, either the cell will destroy that extra thing that can lead to harmful effect to maintain the homeostasis, or there could be some other phenomena or other parakine effect that could happen that can lead to homeostasis of the cellular system. So that means the binding and its related biology we will try to study in our future lectures. Thank you.